Hi guys. So in this video I wanted to talk about how ATP can be produced from glucose in your cells. So in your cells you have mitochondria, and these mitochondria are organelles, so basically parts of your cells that produce ATP from glucose via cellular respiration. So cellular respiration is basically the whole process that we're going to be going over. And it is the process of a glucose turning into an ATP. Now keep in mind, many glucoses are turning into ATPs at the same time. So yeah. And by the way, sorry, but glucose turning into ATP is not actually accurate. It's more glucose is being, the energy from glucose is being used to produce ATP. But we'll see what that means in a minute. So there are three basic sort of stages to cellular respiration, and I call the first one is called glycolysis. And I'm not going to talk about all the names, but it basically is a process that results in glucose being converted into pyruvate. Okay. And also, I should also acknowledge that some ATP is produced, but not a lot. So most of the ATP produced from cellular respiration is going to be talked about later. This is just a small amount of ATP that you get as sort of a bonus from glycolysis. So you have glucose and it's converted into pyruvate. Alright, that is your first step. And there are lots of stages that glucose converts into in order to get to pyruvate, but we're not going to be going over that right now because there's not really much value in memorizing all the names if you're just looking to understand the process. But glucose is converted into pyruvate. By the way, if you want a better like a more detailed understanding on this, you should go to Khan Academy because they have the whole process. The process as an entirety, all of the details, all of the molecule names. So if you're interested in that, go check Khan Academy out. So stage 2, Krebs cycle. Now you're going to understand why we had to make that pyruvate out of the glucose molecule. Because you might have been thinking, well, why did we make that pyruvate? Was it just for nothing? Well, it wasn't. So the Krebs cycle is exactly what it sounds. It's a cycle. And it starts with something called citric acid, and it is being converted into a whole bunch of different molecules. So this might be a molecule, and then this molecule will be turned into a different molecule. This is not what they actually look like, by the way, but I'm just drawing them. And anyways, this happens all throughout until the very end when you get oxaloacetate, and that is the last molecule it will convert into. Acetate. Okay. So, oxaloacetate. So why am I telling you only these two names? Well, they're the only ones that are really relevant to what we're going to be talking about. Well, they're more relevant. So basically, this cycle needs to continue going on. And we're going to talk about why this cycle needs to keep going on in a second. But it needs to go, just take my word for it right now, it needs to keep going on over and over again. Now, in order for it to continue going on, oxaloacetate has to be converted into citric acid. Okay? Now, that we need a special component to convert oxaloacetate into citric acid. And you might be thinking, oh, it's pyruvate. But it's actually not pyruvate. But pyruvate is involved. Pyruvate is converted into something called acetyl-CoA. Now, if these names are overwhelming you, don't worry about it. They're not that important to understanding, but just follow me right now. Pyruvate is converted into acetyl-CoA. And acetyl-CoA is going to be used on oxaloacetate to convert it into citric acid. And then the cycle can continue going on and on and on, with the help of glycolysis, which produces the pyruvate needed, right? Okay, so why does this process need to keep going on and on and on? Well, there are certain molecules that are produced when citric acid is converted into these molecules. So, for example, when this molecule is turned into this molecule, maybe, I'm not saying this is necessarily true, I'm just using this as an example. Sometimes when the molecules are converted into the next step, then a form of energy is produced, and I'm not going to give you the names, but certain molecules, I'm just going to write mol for short, but certain molecules are produced and they carry energy. So this might be the molecule without any energy. The star means no energy, and then it might be converted into a molecule with energy. And then I'm going to border it to show that it has energy now. So now the molecule has energy. And there are several types of these molecules, by the way, but I'm not going to give you the names. Khan Academy does give you those names, though, just for simplicity's sake. So certain molecules are produced from some of the conversions of one molecule to another, and then maybe later, like farther down the road, another molecule is produced, and so on. And then more molecules, and then it goes as a loop. And it produces more and more of these molecules that are holding some sort of energy. Okay? So these molecules are holding energy. So how are we going to use those molecules to make ATP, right? Well, your mitochondria has a wall inside it. So I'm going to give a diagram of the mitochondria now. So this is what your mitochondria looks like. Don't worry, we're going to be getting into the final stage in a second. But this is a basic diagram. You can look it up for a better diagram. But basically it has two parts. It has an outside and an inside. And the inside is called the matrix, and the outside is called the intramembrane space. Again, names are not that important. But, so, when you... The reason I'm telling you this is because this wall between these two things is very important for the next step. So for the next step, you will have the wall. So this is a zoom in of maybe this wall. And you have the intramembrane space on this side and the membrane and the matrix on this side. And you have these proteins here, right? And then you're going to use these molecules. So this is molecule, this is matrix. So actually I'm just going to write it out just in case. And these proteins are going to convert this molecule's energy into ATP somehow. But first, let's make a new page so that it's not so crowded. But you basically have this wall, and you have the intramembrane space on the outside, and you have the matrix on the inside. And this molecule, its energy is going to be used somehow. So how? Well, proteins line, I'm not going to give you the names, but again, Khan Academy has those. It's the last time I'll say that. But it has proteins lining it, okay? And this molecule's energy is going to be used, with the help of these proteins, to pump hydrogen ions, positively charged hydrogen ions, to this side, okay? And these are going to get very concentrated, and positives repel. So, because positives repel, they are not going to want to be near each other. And because these don't want to be near each other, they're going to want to come back into the matrix. They're going to want to go back into the matrix. That's the only place they have to go, right? So, ATP synthase, the name's not that important. Kind of see where it comes from though, right? ATP synthase. But these hydrogens are going to come back in via this ATP synthase, and the protein is going to use the energy of this atom coming back, this ion coming back into the matrix, to convert ADP, adenosine diphosphate, not ATP, ADP, and a phosphate, and it's going to convert into ATP. So for those of you who don't know, ADP is basically the form of ATP without any energy. So it basically doesn't have any energy. And then it is converted into ATP with the help of this protein. And ATP does hold energy. ADP does not. So now you have this molecule that the cell can use. And with the help of cellular respiration and the 3 steps, this step is called the electron transport chain. Sorry about that. So ATP is produced from adenosine diphosphate and this protein with the help of this gradient, so this concentration gradient, so these populated hydrogen ions, they want to come back in, and this energy is used to produce ATP. So with the help of 3 processes, glycolysis, the Krebs cycle, and the electron transport chain, the first thing that happened was pyruvate was used, and that helped these molecules be produced via the Krebs cycle. And these molecules were used to make these hydrogens all pumped to the intermembrane space. And these hydrogens did not want to be near each other, so the energy from them going away from each other was used by this protein to produce ATP out of ADP and a phosphate. So I hope you guys found that useful. That pretty much wraps up cellular respiration. There is another process that goes on when you don't have oxygen. Because we did not go into the use of oxygen, but we'll go into that later. But those are the basics of cellular respiration. So that is the main way, well, basically the way for ATP to be produced in your mitochondria in your cells. Thank you.